Welcome, everyone. My name is Jean Swidler. I'm with Genetic ALS and FTD and the Legacy, and we are so pleased to be sharing this webinar today with you on practical updates in genetic testing in ALS. Genetic ALS and FTD and the Legacy is a patient-led organization proud to serve our community. We strive to serve the needs of our community through education and support resources like today's webinar. And we also serve to represent the interests of our community in dialogue with the academic fields that study us, our governments, and the industry developing drugs for us. I would just like to acknowledge some thanks very much to our speakers today, and of course always to the people who participated in the research and the researchers who allowed for the findings that might be discussed at some point in the webinar. And we do want to especially thank our trailblazer level sponsor, Genetics, with whom this webinar was planned. With that, I'll kick it over to Tara.
Hello, everyone. Thank you for joining us today. I finally have enough time maybe to go over all the genetic counseling topics I'd like to in FTD and ALS, so I'm excited to kind of go over those in more detail with everyone. Essentially, just to kind of start, some of you listening in have probably already met with a genetic counselor, but I do want to kind of highlight some things that we offer that are different from other providers, particularly in our ability to kind of break things down into simplified complex concepts. So educating is a big piece of what we offer to patients and families. We will do an in-depth family history. So we'll go all the way up to grandparents, sometimes go down to grandchildren. And we'll provide a risk assessment based off of your family history. And we do kind of discuss the appropriateness of genetic testing, whether it's right for you in your current life stage, and discuss alternatives to testing as well. And finally, we do offer help in decision-making. We practice a shared decision-making model where from our initial about 1 hour long session, we get an idea of your motivations for testing and help you come to the right decision and provide support in that as well. And I know a lot of you have personal experiences with ALS and FTD. MND is another term for ALS. It's motor neuron disease, more commonly used in Europe. But I do want to kind of point out with ALS, it's pretty easy to consider, do you have any family members that have progressive muscle weakness, who may have had a sharp decline? But with frontotemporal dementia, it can be very difficult to diagnose and can look very different in different family members. So it might look like someone going through a midlife crisis and they go out and buy a bunch of cars that they can't afford, just like a major personality change. Executive functioning wise could be something like someone stopping in the middle of the road while they're driving because there's too much to process at once. And language can be affected with difficulties with word finding, saying sentences out of grammatical order. Emotional regulation, inhibition can be difficult to kind of keep tempered down. And we know that the two are related genetically. And so that's why when we're thinking about if you have family history of a similar disease, we think of a family history of a spectrum of diseases. And so if someone has ALS or FTD, should they get genetic testing? Um, and apologies, I didn't update the slide, but for ALS or FTD management, genetic testing might not change your medical management unless you test positive in SOD1. But it could offer the opportunity for gene-specific clinical trials. It is important to realize that even though we're testing you, this applies to other family members as well who could now be at risk if you do test positive. And then regarding insurance coverage, it's a bit of a moot point for genetic testing at this point, because currently there are several options for free genetic testing via various genetic testing labs, as well as probably Genetic and Light the Way, which we'll go into a little bit more detail later. And then I kind of want to point out what genetic reports might look like. So this benign or likely benign category is probably not going to be listed on a genetic report. These are variants that don't appear to have any consequence to the gene product. Whereas pathogenic and likely pathogenic will definitely be on the report and uncertain will be on the report. We're no longer really using this term mutation, but we're calling it an uncertain variant or likely pathogenic variant. So just realize that on your reports, it's no longer going to say mutation. And typically mutation used to be just referring to likely pathogenic and pathogenic variants. But to go into more detail on these uncertain variants, which I think is important, it essentially is a change in the gene that isn't seen often enough in the general population. So in other words, you know, it could cause the gene to not work, or it could just be a unique difference. So imagine you can say color with an O, you can spell color with an O and a U, and it still sounds like color. That could be a truly benign variant that has no consequence. And so it will take important consideration and interpretation by a provider that's knowledgeable in genetics to give you an idea on how suspicious we should be of some of these uncertain variants. And so if you kind of decide to do genetic testing, what are the chances to have a pathogenic variant or test positive? Specifically, we're talking about ALS in this scenario, where if you do have family history of ALS or a related disease, there's about a 2/3 chance to test positive. Whereas if you are a completely sporadic case, we have no family history, there's still up to a 10% chance to test positive. This is most often due to the most common hereditary cause of both ALS and FTD, which is C9. But if you do end up testing positive in a sporadic category, we might actually now consider you to be familial. A lot of these sporadic cases might just appear to be sporadic because family members either died young or we had incomplete family history information. And then on the other hand, if you have FTD, chances to test positive is about 42% in familial cases, whereas in sporadic cases, it's about 12%. And I do want to note some of the complexities of dementia genetics in particular. So we have a clinical diagnosis of a specific subtype of dementia. In these cases, we've got Alzheimer's, frontotemporal, a dementia with motor symptoms. So that could be like cortical basal syndrome. and prion disease. And you can see that there's kind of an intermixing of genetic causes. And what I'm trying to get across here is if you think you have a familial form of FTD and you only tested for the 3 common FTD genes, C9, progranulin, or MAP-tau, maybe that genetic testing should be expanded upon, and we should be looking a little bit more broadly. And finally, I do want to touch on DNA banking. This is an alternative to testing for people who don't want to move forward with testing. Or if you do think you have a familial case, but you don't test positive with the current genetic testing available, as well as if someone is considering in vitro fertilization with pre-implantation genetic testing to prevent disease from passing down to future generations. In many cases, it will be beneficial for both parents of that person to have DNA tested in order to kind of improve the likelihood of success in the procedure. And then I just kind of bring up a website for some DNA banking options. Unfortunately, this is not a very affordable option, but this is a trustworthy lab, the Center for Human Genetics. They've been around for about 40 years. So certainly talk to your genetic counselor or neurologist about DNA banking if it's something you're interested in. But Prevention Genetics is no longer accepting new samples for banking. Okay. So considerations for asymptomatic testing is something that, you know, takes a lot more thought. I've reached out to the community to get some input into how I present this slide. And together, we've kind of come up with this self, family, and relationships and society categories of things to consider. So to break it down, essentially, I usually say if genetic testing is going to change anything about your future, maybe it is right for you. So by career and travel planning, I mean that there's a possibility that you might decide, I want a desk job, or I want a job that's going to keep me more active or use my brain more. And via travel planning, you might decide, I'm not going to wait till I retire to do all of my big trips. I'm going to kind of intersperse them throughout my career. Financial planning, of course, it can give one a better sense of control. So it can increase the opportunity for medical surveillance that can lead to an earlier diagnosis, possibly earlier entry into clinical trials. It can improve one's sense of self-advocacy for themselves. People might go out and take efforts to try to prevent disease based off of some scientifically sound ideas, such as exercise, for example. And it can also help to relieve anxiety. You can imagine not knowing whether or not you carry the genetic risk factor. At least by testing, you can hopefully remove that uncertainty, but I do highlight that there still ends up being a bit of uncertainty in cases where someone does test positive. Family and relationships category, It can clarify the risks to your descendants. There is a concept of parental guilt that is a very real feeling, even though we know that we don't choose which genes we pass down to our children. It can affect someone's family planning. I highlight survivor's guilt here. That often is in terms of with siblings. So if one sibling tests positive and another one tests negative, the one who tests negative, it might actually be bad news for them. They might feel that they want to kind of take that risk from their sibling. It can affect, you know, you might actually think about who you want your support person to be when you get tested. You might consider, I don't want to bring a support person who is personally at risk of this disease, or I'd rather not bring my spouse because it's going to really affect them when they hear the results. And I'd rather bring someone a little bit farther removed for this test result. And then, of course, it can affect your current and future relationships. And then finally, in the society realm, it can kind of bring you to community, but sometimes you don't need testing for that. It can increase your sense of activism. It definitely can improve your opportunities to participate in research. But do keep in mind that for observational research, a lot of times you don't need to know of your own genetic status. Genetic discrimination is a topic that we will discuss in a future slide, and then cost of testing, particularly financially, can be something to consider for your own testing. Okay. Family planning options, I highlight in alphabetical order. If you know the familial variant, if we know the cause of disease in your family, adoption is an option. Egg, sperm, or mitochondrial donation is an option, but those prevent you from having your own biological children in some cases. And we did touch on in vitro fertilization with pre-implantation genetic testing, as well as there's an option of natural conception and then testing prenatally, although it is only advised that you test the pregnancy if it's going to change the outcomes of the pregnancy. And as well, you might not factor your genetic results into your decision. And I did want to really touch on genetic discrimination, which is the means of using genetic information to limit a person's equitable access to something. And this is particularly important if you don't show signs of the disease. And a lot of times when we think of genetic discrimination, we think about obtaining certain insurances. So this applies in the United States, where we have a Genetic Information Non-Discrimination Act. And it protects health insurance and employment for places that have at least 15 employees. It does not protect life, long-term care, or disability insurances, and it does not protect for certain military or governmental insurances. This genome statute and legislation database on the NIH website will point you towards any specific state protections that were either passed or never went through the legislative procedures. And then just to keep in mind, this is what we think about when we think of genetic discrimination, but we really can't fully anticipate what genetic discrimination might look like in the future. So keep that in mind. That certainly is a big factor to consider. All right. And finally, if you do decide that you want to get tested, there's a big difference between deciding to get tested clinically versus research-wise. If you do clinical genetic testing, you do get a report with your name and date of birth on it, and you often get results quickly. Whereas on research, you usually have a study ID, which often does provide more anonymity. But the research results can take a very long time. Clinically, you would need a report with your name and date of birth on it if you want to get any sort of insurance coverage for the IVF with pre-implantation testing. And with research testing, there's a greater chance that the results are inaccurate. And this is still a possibility even with clinical testing, but you can imagine if the test results are going through with the study ID, there's just an extra element to consider about whether those results would be accurate. And with research testing, you do often need a strong family history or a documented familial variant to be eligible for some of the research studies. Here I highlight some of the research studies available for ALS, and all FTD is one for FTD. And then if you do decide to do clinical testing, there's a website, findageneticcounselor.com, where you can kind of check off ones who specialize in neurogenetic conditions. And if you do want a little bit more anonymity, you might ask your genetic counselor if it's possible to use an alias or a shadow account. But that still might be linked to your actual name and date of birth. So when we're thinking about, do we want our information to remain anonymous, it's still important to reach out to the study coordinators and ask what information might be entered into an electronic medical record system, and could that interface with outside institutions? There is a new program called Light the Way for people with ALS or family history of ALS that offers free testing. And that is kind of kept separate from an electronic medical record system. And it is still clinical-based testing that offers a report with your name and date of birth on it. However, it also gives opportunities to participate in research at the same time. There's also, for people with FTD, there's a program called Probably Genetic that is kind of patient initiated, where you go to a website and you answer a few questions. If you are determined to be eligible for the testing, they do offer testing through that program. And again, that's another kind of clinical-based testing that's kind of separate from the electronic medical record system. But always, if it's something that is concern to you, make sure to ask questions. All right, and that does conclude what I wanted to discuss. And I just wanted to end on this quote, "You never know how strong you are until being strong is the only choice you have." I think that applies to a lot of our families with ALS and FTD. And just keep in mind, don't let anyone pressure you to get testing. Just make sure it's right for you. All right, thank you. I will pass it off to Amar.
Thank you very much, Tara. I'm just going to share my screen. So I'm going to be talking about the research aspects of genetic testing in ALS and FTD. and the importance of research, why genetic research particularly can bring hope. And I noticed one of the questions was about why research testing might be less accurate than clinical testing, and I'm happy to address that after the talk. So the reason why we need genetic research is because we need to answer the question, how do we find a cure? And the way we find a cure is we hunt for drug targets. So drug targets are body systems that can be influenced by a treatment. So if you have a drug target, then you can find a treatment that might affect that and therefore impact the way the disease manifests. And the way we find drug targets is by understanding what causes, in this case, ALS or FTD, and understanding how that problem evades the body's repair systems. Why do people develop ALS, but then the body can't fix it? So to do that, we have to understand what causes ALS or FTD. And there are really only three possibilities. It's either going to be a genetic basis, because genetics drives how we are designed, the exposures that we experience during life, because those interact with the genes that are expressed in our bodies, and then it could just be completely random or a result of time and wear and tear. So it's really one of those three things that's likely to be underlying any neurodegenerative disease, but particularly ALS or FTD. And of those three things, although they're all challenging to study, genetics is the least challenging. It's the easiest to study reliably. When I say the easiest to study, it's still a huge, very, very difficult task. For example, the human genome is 3 billion base pairs long. Each base pair is equivalent to a letter in a code. And when we're looking for a variant that might be causing disease, we're looking for one change in a sea of possible changes. So it's a very challenging thing to achieve. So what have we learned from the genetics that has been done so far? We know that genetic variation contributes about 60% of the risk of ALS on average. And we know that from twin studies, because identical twins share all of their genetic information, but only half, and all of their environmental exposures, at least in early life. Whereas non-identical twins only share half of their genetic information, but do share all of their environmental exposures the same as identical twins. And so by comparing how often are identical twins identical in disease compared to non-identical twins, we can understand how much of the risk is genetic versus environmental. And if we do that, we can show that the risk for ALS is probably about 60%. The contribution of whether you get ALS or not is probably about 60% from genetic variation. That doesn't mean the genes are big genes that are really making you definitely develop ALS. They're things that may just nudge you towards getting ALS. So just having that variant just slightly increases your risk, maybe by 1%, 2%. But if you've got enough of those, then it may take you over the threshold. In some people, the specific genetic variation they inherit actually greatly increases their risk. And so in those people, there may be a family history because other people carry that same genetic variation that is greatly increasing the risk. So it's different from variation that just nudges you towards developing ALS. And about 10 to 20% of people carry one of those variants. So I know Tara said it was 10%, and the reason why there's a slight discrepancy just comes down to how you define what you mean by increasing risk. So this is a study actually that Tara referenced on her slide. And if you look at the y-axis, we've talked about the probability of having a clinically actionable genetic test result. So that doesn't mean that the result will definitely cause ALS or FTD, but it does mean that it's something that will need to be discussed with the clinician who did the test. So 10 to 15% of those results will be ones that we would regard as definitely or likely to cause the condition. But some of those variants, so maybe another 5 to 10%, would be variants of uncertain significance. So those are ones where we're not absolutely sure they cause ALS or motor neuron disease or FTD, but we think that they may well do. So those are the ones that are quite difficult to work with because there's uncertainty both for the person who's receiving the result and for the person who's trying to give the result. What you can see on this graph is that if you test at different ages, it doesn't really make that much of a difference. We did think the reason we did this research was because we did think before that if you were younger, you were more likely to have a positive genetic test result. And in fact, in the UK at least, our genetic testing was predicated on only being allowed in people under the age of 40 if there was no family history. So following this, we've now been able to test everybody. So how does genetics help? Well, we know from all the genetics done so far that the genes that seem to be involved in ALS fit into 3 broad categories. Those categories are genes that cause that affect the nerve cell skeleton. Those are called neurofilaments primarily, but there's lots of other components to the nerve cell skeleton. They're genes that code for proteins that are involved in making other proteins or in components that make other proteins in the cell machinery. And finally, they're genes that are involved in the kind of the garbage disposal, the rubbish disposal in the cell. So the cell cleaning systems, what we call the proteasome. So those 3 categories of genes seem to be broadly involved in ALS and FTD for some reason. Some genes also influence disease progression because, of course, just because a gene causes the disease doesn't mean that it's going to make the condition progress. So a nice analogy is to consider if you are lighting a bonfire. If you set fire to a bonfire, which has maybe gasoline on it, so you ignite your match, you light the bonfire is blazing, that's like disease onset. Now you blow out the match which caused the bonfire, it doesn't make any difference to the bonfire itself. So genes that are involved in disease onset aren't necessarily the same as the genes involved in disease progression, but they may be. In the same way as the gasoline would obviously be required for the bonfire to start, so would the wood. And they also, of course, affect how the bonfire will blaze. So genes affecting disease progression also make good drug targets. And the reason they make good drug targets is because people come to us, in most cases, after ALS or FTD have begun. And so if we know that it's already started, we need to know what are the targets that can then influence the disease from that point on. This is a graph showing the genes discovered since about 1994 when I started in ALS. So SOD1 was discovered in 1993. What you can see here is several things. The first is that the graph seems to be rising exponentially. So roughly every 4 years, the number of genes discovered doubles. Another thing you can notice is that the size of the spheres differs. The bigger the sphere, the more publications there are on that particular gene, and that suggests it's more important in ALS. So SOD1, C9orf72, which is a very big sphere in the middle, TARD-BP, which codes for a protein called TDP43, that seems to be important in nearly all of ALS, and FUS, which is a related protein. Those are the 4 big ones. What you can also see is some of the spheres are blue and some are red. The ones that are red are variants that have only ever been found in people with no family history. The ones that are in blue are the ones that have been found in people with a family history. And you can see that most of them are found both in people with a family history and without. The other thing that you can tell from this graph is the color coding of the actual labels. So the blue labels are for genes involved in the nerve cell skeleton. The red ones are involved in protein making machinery, and the green ones are involved in the cell cleaning system. So you can see the 3 broad categories of gene. Now this graph is only up to 2017. There's many more to add. I've just taken it from a publication we made back in 2017. The same principle still applies. Now, I did say that some gene variants affect the rate of disease progression. Actually, even within a diseased gene, you can have that effect. So these are some of the variants found in people carrying variants in the SOD1 gene. So along the bottom, you can see the different variants. This is just technical terms. So the first letter is the normal amino acid found at that particular position. So they're numbered. And then the second letter is the amino acid code for what it's changed to. So if you look at A5V, which is the 3rd one along, that is at position number 5, and alanine has been changed to a valine by the variant. And you can see that people carrying the A5V variant are actually, or the dots cluster, the y-axis here, the vertical axis, is the disease duration you can expect if you carry that disease variant. So you can see that people carrying that tend to have very aggressive disease. It's very rapid in progression. Whereas if you move along to the right, you can see the ones carrying D91A, that variant is quite common in Scandinavia. They have a much longer disease progression on average, so typically more than 5 years. And then there are some variants like on I114T, where some people have very aggressive disease and other people have very slow progressive disease, and there doesn't seem to be a relationship. So you can see that even within a single gene, you can have variants that have different effects on disease progression. So how does genetics actually help? Well, there's a direct impact of gene discoveries. Apart from the genetic counseling and testing and all the implications of that you've heard about from Tara already, there are also the possibilities of treatments using genetic therapies. So we've only got one genetic therapy so the picture there represents the single treatment we've got in the huge desert of treatments that are needed. But we have got a therapy now called tofersen, which targets people carrying the SOD1 variant. It targets particularly the SOD1 gene and manages to slow down the disease dramatically in many cases, and particularly if given early enough. So that's a really huge advance for ALS. Only 2% of people carry that particular gene variant, but that still means we have a very effective treatment for 2% of people. And that's not something we could have said until very recently. So here you can see some graph results. So the initial short trial of tofersen didn't seem to show a clinical benefit, but the trial included measurements of a protein in the blood called neurofilament. And neurofilament is part of the cell skeleton. And when nerve cells are unhappy or dying, they release neurofilament, and that passes from the spinal fluid and the brain surrounded by spinal fluid as well, even though it's called spinal fluid. That leaks into the blood, and then we can measure that in the blood. And we could see that in people on this drug, on tofersen, their neurofilament levels were dropping about 3 months after taking the medication, even though there didn't seem to be a clinical effect at that point. But then when the people were followed out further, you can see from the graphs on the right, the top panel shows the ALS functional rating scale, the total score. You can see the people who had the drug to start with, so they started with the active treatment. They had a good response. The people in the blue arm, they had a delayed start. So they didn't start until 3 months after the initial dose given. So they had a placebo until then. And they still showed some slowing, but it only had benefit from the position they were at. And then if you look at the respiratory results on the lower panel, you can see the same kind of pattern. So that suggests that tofersen really does have a clinical benefit, and that the neurofilament levels did suggest that that was going to be there. You can also have an indirect impact from understanding genetics. For example, we can understand the disease and we can design new treatment based on that. We can also stratify people in clinical trials, because if we think that a particular gene or genetic background or genetic pattern means that the disease is caused by a particular combination of events, we can target those particular drug targets specifically. Excuse me. So here is an example of that. What you can see on the right is people carrying a particular genetic variant in a gene called UNC13A. And if you carry that genetic variant, you have a very aggressive form of ALS. And you can see that in the pink line there. Those are people who have that genetic variant and were given placebo. Whereas the people who are in green, they carry the aggressive variant, the poor prognosis variant, but they were given lithium and they responded to the lithium. So it didn't stop the condition by any means, but it's changed it from an aggressive condition to the same pattern as everybody else. So that's a way that genetics can help even with traditional types of therapy. And genetics could help as well, potentially by predicting onset or preventing onset. That can only be done in a targeted way. That's a property of statistics. So we can't screen the whole population. I'm happy to discuss that with people if they want to know why, but it's purely a property of the universe. It's a statistical issue. Genetic testing in people with increased risk is possible, and that would usually mean people in whom there's another family member with a genetic background, or in people who've tested positive themselves. And there is a trial at the moment doing exactly that called the ATLAS study. that looks at the neurofilament levels in the blood and then gives tofersen, if the person carries SOD1 variants, it gives tofersen to that person as soon as the neurofilament levels rise. So just to summarize, understanding genetics helps because it helps us understand the causes of ALS, helps us design new genetic therapies, helps us design targeted treatments that are more traditional in design, and it helps us identify who is at risk and potentially allows us to prevent ALS onset. So thank you very much. I will now stop sharing and hand over back to Jean.
All right. Thank you so much, Amar. Very, very helpful. I plan on watching the recording again because I was managing some of the questions. But I'm gonna take over the Q&A for Jean because she's having some internet difficulties. Someone asked, do you have any recommendations on how to or whether to let our family members know about our results, including cousins, aunts, and uncles? I think that's a great question for me to answer about family communication. And obviously, the more distantly related you become, or you are, I guess, the harder it is to know, do you just reach out to that random cousin that you haven't seen since your reunion a few years ago, or even cousins that maybe you've never connected to in the past? It's always generally recommended to share this information with all at-risk family members. And sometimes if you start with the generations above, if that person tests positive, then the direct descendants of those individuals now know that they're at a 50% risk. We call that cascade screening, but sometimes the parent doesn't want to get tested and the children decide to go out and get tested regardless. And you can imagine that will potentially reveal the genetic status of the parent. So these are all things that genetic counselors talk in great detail with patients about. One thing that genetic counselors often offer is a family letter to share this information with relatives. And it can be done in an email to those individuals with the letter attached saying, hey, like this is important information for you regarding so-and-so's history of such and such disease. Feel free to read it when the time is right. So that kind of tells them a little bit about what's in the letter, and people can choose whether they want to read it or not. It's tricky, but it's still advised to share that information. And one of the biggest reasons is because sometimes people already know there's something going on. And if they know the genetic cause, they know this is what I test for, and it gives them the greatest sense of control. So you can imagine this information can be daunting to people and it might hit them really hard, but eventually most people do learn to cope with that knowledge. So I hope that helps. Do you have anything to add, Amar?
No, I thought you were very comprehensive. Very good. Thank you.
Okay. Let's see. Amar, you mentioned you wanted to answer this question. Why are research results less accurate? That's kind of scary when we think of the importance of research.
Yes. So it's not necessarily that the test in the laboratory is less accurate. It's just that in a diagnostic laboratory, it's certified. So it goes through a lot of quality control. And the tests they use make sure that there's very little chance of any clerical error. You can never reduce that to zero. But because of that, the result coming from a diagnostic laboratory has a very, very high certified qualification of being accurate. The work in a research laboratory doesn't need to go through that because we're answering the question for ourselves for research. And certainly in the UK and in some other countries, you're not allowed to give the result back to the person because the actual sample is completely anonymized. It's separated from the person who gave it, so we can't return the result. So in that sense, the clerical errors that might occur are really that the ID is mislabeled. And if that happens, it's unlikely. We know it's unlikely because we do checks of the sex of the sample compared to the sex reported by the person. So we know how often there's a mismatch. That's quite a good quality control check. So we know it's unusual, but you can get clerical errors in research studies. So that's why they're less accurate. They're not certified at a high quality. It doesn't mean they're not accurate. They're just slightly less accurate.
Great, that's a wonderful clarification. And yes, another thing with research testing is they'll often run all the samples in batches. So even if it's not truly anonymized, it does increase that risk. Okay, another question is PGT, that's pre-implantation genetic testing, available for all ALS genes or only some? That is a really great question. I'll try to answer it as best I can. It's going to be genetic counselors specializing in assisted reproductive technology, or ART, who will be able to best answer the question. But generally speaking, if we have a known familial variant that is pathogenic or likely pathogenic in a gene that has a very clear disease association, things like genes like FUS, like TARD-BP, MAPTau, C9, SOD1, that is likely going to be available to do PGT on. If it's something that's an uncertain variant or it's just a susceptibility for ALS, it's going to be dependent on the genetic testing lab that tests the embryos, and they kind of get to decide whether they'll accept a case or not. Another piece is some of the repeat expansion disorders are a little bit harder for the labs to really test for it with a single cell from a developing blastocyst. So that might be another case where they'll require grandparent samples to compare to, because they won't be able to truly test the expansion directly. So it's complicated, but most genes should be available, but it is considered on a case-by-case basis. All right. With all of these options for genetic testing, is there a mechanism for all participants to opt that their data be included in a big informative pot of ALS genetic data? Can this data be combined with projects like is it MINE or MIN-E?
Project Mine. Project Mine.
Go ahead, Amar.
Okay, I can answer some of that. So certainly in the UK, we do have that option. When somebody is tested diagnostically, there's an option on the form where we make the request to ask if the person would like to give their sample anonymously for research. If they say yes, and that's not just for ALS, that's for any genetic test. If they say yes, it goes into a national project called the NIHR Bioresource, and then that goes through whole genome sequencing, and that data is available to researchers to download and use.
All right, thank you for that. Do you have a sense for repeat length in C9 and age of onset? In other words, is there any documented anticipation in FTD? Do you have the answer for that, Amar?
Yeah, I can answer that. I can talk to that at least. So C9 is slightly different from usual repeat expansion. So most repeat expansion diseases, repeats, by the way, are small sections of DNA. So there may be 2, 3, 4, 5, 6 letters long that are repeated multiple times. And for C9, it's 6 letters, GGGGCC is the code. That's repeated just twice in most people, but there is some variation. So some people have 5, 10, 20 repeats. We know that if you have more than about 28 repeats, then you're at risk of ALS FTD. But most people don't have 28 repeats or 30 repeats. Most people who have an expansion have tens of thousands of repeats of that. And so really, when you're getting to that size of repeat, it's very difficult to measure it accurately. And it may even differ in different cells in the body. And so we can't really do the research to answer very well yet, does the repeat length actually affect your age of onset for ALS or FTD for C9, just because it is so variable? We can tell that if you have an intermediate sized repeat, so 28, 30, something like that, it still gives you the same kind of risk as if you have a 100 repeats or 1,000 repeats, but we can't really tell much more than that yet. Did you want to add anything to that, Tara?
There's actually a couple of questions on both SOD1 and C9 penetrance. And so I think it is important to note that originally the families that first helped us discover these genetic causes were highly penetrant families where there were many generations of ALS and or FTD. And so when we start to look at people prospectively, where maybe we identify a single sporadic case of ALS in the family, or we start doing population-based screening, we learn that the penetrance of these conditions are lowering from what was originally quoted. And that is very important. And then to think, you know, if you've never seen FTD before until, you know, years after something strange was going on with a family member, and then finally we get the diagnosis, now people in the family might be hypervigilant and they might kind of notice subtle signs or symptoms sooner, where they could still come to a neurologist and not meet a clinical diagnosis. So those are important things to consider, with FTD being very difficult to diagnose. There's another question about penetrance of SOD1, and depending on the genetic variant, the penetrance might be greater or lower. And we're seeing a lot of variants of uncertain significance in SOD1 where it might be uncertain because the penetrance is so low, and it might actually be very difficult to determine if we find this variant present in a much older unaffected relative, does that actually give us enough clues to say whether it's likely benign or likely pathogenic? So do you have anything to add to that?
Please, yeah. So there's two things I'd like to add. So the first is that there is an ongoing initiative run by ClinGen, which is part of the National Institutes of Health in the US, where there's a variant curation expert panel, a VSEP it's called, where a number of us meet on a regular basis, and we're going through every single variant in SOD1 to decide what is the evidence that this is pathogenic, or should it be considered a variant of uncertain significance, or are there any variants in SOD1 where there's no real evidence that they're relevant to ALS? Maybe they're just coincidentally happening. So we're addressing all of the evidence for every single variant. There's more than 150, so it's quite a lot of work. And then your comment about C9 penetrance, I fully agree with. So penetrance is the probability of developing a condition if you carry the gene variant. When we were first discovering the C9 gene, I had a pedigree from my colleague Peter Anderson in Sweden that had more than 200 members. And in that pedigree, the chance of developing ALS or FTD was about 1 in 10. And we now know that pedigree is a family that has C9 in it as the cause of their ALS or FTD. So that tells you that carrying that gene isn't necessarily enough to develop ALS or FTD on its own. There's probably other things you need. It does greatly increase your risk, but it's not 100%.
Great, thank you. And someone else had mentioned that their genetic counselor had told them that the risk with C9 is basically 100% to develop ALS or FTD. So we can very honestly and clinically mention that 100% should be dismissed completely, but it will still be very dependent on your family history. All right. Let's see. Another question we have that I think came from people asking questions when they registered, and this might be our last question. Is there any research about whether healthy lifestyle or nutrition can affect gene activation?
I don't know of any research that specifically looks at that. Measuring gene activation is quite a difficult thing to do. You measure, there's a few ways to do it. So we can measure a thing called the transcriptome, which is which genes are switched on and off and how much they're switched on and off. But it depends very much on the cell groups you're testing. So you can do it in a single cell, but then how relevant is that for all the cells around? And if you do it for a number of cells, then you're not answering the question for a single cell, etc. So it's quite difficult to interpret. There's another way you can do it by looking at a molecular switch in the DNA called methylation. So there are ways you could do this, but it's a very challenging thing to do, mainly actually not because of the gene switching on and off aspect, the gene activation, but more because measuring healthy lifestyle and nutrition over a lifetime is very, very challenging. And it relies on recall more than anything else, on questionnaires. And those aren't usually available until after somebody has a genetic test.
Great, thank you. All right, so I think we can pass it off to Lindsay with Cyanogenetics.
Thank you both so much for your presentations and for everyone who's listening. I just wanted to flag that we do have some sponsored testing programs that are available both for ALS and FTD. They're available on the website along with many other programs. So obviously, we think you should consider all of your options. What we do at Sano is direct to individuals living with a diagnosis or those who may be concerned that they have a gene and would like to find out. So there is no physician order, but you do have access to pre and post test genetic counseling. So the page on the website is for the genetic testing page. And then we just wanted to say a huge thank you to the End the Legacy team for all of your incredible partnership over the last few months, helping us get the program up and running. We are beyond grateful for your support, and obviously for both of you, Tara and Amar, for your support as well.
Thank you.
All right, we have 3 more minutes. Let's do one more question. Is there anything in the US, like the UK, opt in for sharing genetic data for research? If not, how might we do that? You might have a better idea, Amar.
I'm not aware of anything like that in the US, but actually you can set it up. So it may be that some clinics, for example, MGH may already be doing this, or Mike Benatar's group in Miami. There are ways you can do this because each country has its own legal framework for the difference between genetic testing diagnostically and genetic testing for research. And it may be that you have to take 2 different samples and consent twice, but one then goes into the lab and the other goes off to the diagnostics. Or it may be that you can just tick the box and then it's allowed for both. So it should be possible, even if it's just on a local level, if you ask the researcher or the clinician who's looking after you.
Yeah, and if I recall, there are a couple of NIH initiatives for patients to share their genetic reports in research. And a lot of those will be listed somewhere on your genetic report, but it's not really emphasized. So read through those carefully. Reach out to the genetic lab that did your testing to see what options there are. I'm sadly blanking on the name of some of those initiatives, but that could be a great way to kind of share your data outside of from within the internal genetic lab. All right. Okay, so thank you very much to everyone for participating in this webinar and attending and giving your full attention. I will double check with Jean to see if we have any opportunities to answer some of the unanswered questions if anyone did share their contact info. So we'll do our best to answer those. And stay tuned for more events from End the Legacy.